Everyone. Yeah, I'm so glad there's still several people still here. So my name is Guangyu Wang. I'm assistant professor in Houston Methodist Hospital, and I'm also a joint faculty in Weill Cornell Medical School. Today's topic will be focused on how to use single cell to infer the cell-fate transition. Uh, so this is a study we published last year in Nature Biotech. Um, so actually, I have studied cell-fate transition for a long time. In last time, so before we have single cell technology, we majorly use bulk RNA-seq and a lot of epigenetic technology like ATAC-seq and histone modification for different H3K4 tri or H3K27 tri methylation. But after single cell technology was developed, I think it became a perfect tool for us to study cell fate. So in our human beings, so our human body was built up by 30 trillion cells estimately, and we have many cell types. In every cell type, there is a lot of subcell type. But the amazing thing is, all this kind of different cell type was developed from one single embryonic stem cell. But why we can so well organized to develop our whole body from one cell? What is the signal cell received to regulate the cell fate? It is a mirror for a long time, and if we can understand how cell fate was regulated, so actually we can treat a lot of disease. For example, heart failure. In heart failure, we have a lot of fibroblast cell generate from somewhere. So actually we don't know why. We know partially the results, but we don't know exactly where the fibroblasts come from. Maybe the fibroblasts come from the proliferation of fibroblasts. Maybe there is transdifferentiation event that fibroblast cell was generated from endothelial cell or cardiomyocyte. And if we can understand how we can regulate the cell fate, maybe we can regenerate the failure of the heart without transplant. This is one example. Of course, there is a lot of examples. For example, if we understand why cell becomes the senescent cell, maybe we can restore the senescence. So there are many applications. And actually, cell fate, for a long time, we think for human beings, the cell fate was fixed. Actually, it is not. We already observed a lot of transdifferentiation event, and also we can reprogram cell back to iPSC stem-like cell. But actually, our knowledge and understanding is quite limited. So our study try to upgrade this kind of low resolution, low dimension, old type cell fate landscape to the high dimension, high modality cell fate, and highly accurate that can be used for us to maybe help for the precision medicine. But it is very difficult. It is because the cell fate is not regulated at one layer. So our cells was regulated at so many layers, like the epigenetic regulation, translational regulation, post-translational regulation, and also at protein level, we have different type of modification and regulation. Protein has different degraded speed. And so several years ago, there is a Nature paper published named RNA velocity of single cell. So they try to claim a new framework for us to understand cell fate from single cell data. So their model is a two-water pool model, this kind of water pool. The first one indicated unspliced mRNA. The second one is spliced mRNA. So at beginning, after mRNA was generated from the transcription, so actually it is unspliced. Both intron exon are there. And the next step is splice the pre-mRNA to the mature mRNA and add the poly-A tail and the 5' cap. So there is a delay that mature mRNA was generated. So from every cell, actually, we can capture pre-mRNA and mature mRNA at the same time. So in this Nature paper, they propose a very simple ODE. So this kind of ODE equation that can describe the dynamic of how the mature pre-mRNA become to mature mRNA and how the mature mRNA degraded into the fragment. So actually in this ODE, there are 3 parameters. The first one is alpha, indicated the transcription rate, and beta indicated the splicing rate, gamma indicated the degradation rate. Okay, so let's take a look at the real data. Whether this equation is too simple that cannot reflect any truth of the cell, or it is, although it is very simple, but it is still the truth that we can use it as a principle that to understand the cell fate. Actually, the same Nature paper gave us a very, I think, a very interesting example. They just simply use the bulk RNA-seq data to study the circadian during a day. So you can see in this figure, there is different time of a day. So, and here is a time point 0 on splice rates, and here is the splice rates. You can see the splice rates at time point 3 is very similar to the unspliced rates at time point 0. And if you carefully look into this graph, every next time point, the splice rate is very similar to the previous time point's splice rates, unspliced rates. So U indicated unspliced, S indicated spliced. You can see splice is very similar to unspliced as the previous time point. So this is the data suggesting maybe we can use unspliced rates to predict the future of the whole transcriptome. So if you look at one gene, CBS, this is very famous circadian gene, you can see it forms a loop. So the y-axis indicated unspliced rates, x indicates spliced rates. It forms a perfect loop. So you can, and this loop actually follows the solution of this ODE. So if you put all the genes together into using the principal component analysis, you can see the previous time point can predict the future of the differentiation direction. And if you apply the same technology to the single cell data, because this is bulk, so if you apply to the single cell data and train a very simple random forest model, which is this Nature paper used, you can predict the future of the differentiation in a single cell data. So then after I start my lab, so I think maybe we can upgrade their algorithm to a deep learning framework. So why we think it can be upgraded? Because in the original study, they claim 2 phases of the transcription. The first phase is shutdown phase, off phase, and the next another phase is on phase. So they separate the same equation into 2 phases. And I think maybe we can use a sigmoid function to simulate the same function. And sigmoid function is one of the most famous active function in the deep learning framework. So I think maybe we can use deep learning to predict the cell fate. So how to predict the cell fate? It's very simple. So for every cell, we can get the splice rates from intron region and unspliced rates from exon region. So every cell we have the S and the U, and we put it into a very small, fully connected neural network, and the output is alpha, beta, and gamma for this cell. And then we have alpha, beta, and gamma, and we also have S and U. So using alpha, beta, and gamma, and S and U into that equation, ODE, we can predict the future of S and U. Then we will calculate a loss function, which is a cosine similarity, to calculate our prediction and the real data. You can see before training, there is no pattern, so we just randomly predict something. And after the training, we can really predict the velocity of this gene. the y-axis is unsplice, x is x is spliced, y is unspliced. So you can see the gene's expression increasing, and at this time point, the transcription is shut down and it starts to decrease. But the unspliced rates always move ahead of the spliced rates. So we propose this model, which we call it relay model, because we predict every cell's velocity and then relay them together. So we call them relay model and published in Nature Biotech last year. Now I want to give you several case studies. So what is the application of our algorithm? So what is the unique things only we can do, but not other algorithm for the R velocity? So I will give you three major topics. The first one is how to predict the cell phase transition and trajectory, and then how to predict the transcription, splicing, and degradation rate for every cell, and how to predict the pseudo time, and how to in silically perturbate the transcriptome. So this is a case study we download from a Nature paper. So this is the mouse hippocampus data. So we already know, okay, from the pre-knowledge, we know here are two types of progenitor cell. This cell will, some of them will differentiate to this neuron cell, and some of them will go to this another kind of progenitor cell, and some of them will go to astrocytes. So from literature, we already know there are 5 different directions of the differentiation. But if we don't apply R velocity analysis to this data set, we will know, okay, they are the same cell, they are the same cell, there is a cell population, but we don't know the dynamic of the cell. So using apply our algorithm, we can say we predict this is a source of the differentiation. They will different into 5 different types of cells. And for example, this is interesting example, DCX. Actually, this is a marker gene of the neuroblast, highly expressed in neuroblast. So the x-axis is a expression, our normal expression. Y-axis is a new layer. Only R velocity have this layer, unspliced rates. We can see combine unspliced rates and spliced rates, we can see a perfect dynamic. So the DCX expression increase, and at this cell type reach the steady point, and then start to decrease after this neuroblast, because the neural keep differentiation into different kind of neural. This marker gene decrease. But if you look at the gene expression data, the color indicated the expression, you can find some pattern. Okay, this gene will not express in this kind of cell, highly expressed in them, but the information is very, very less than the dynamic information. And another very more important interesting example is PSD3. This is a gene that has different regulation in different differentiation direction. For example, we already know there are 5 different differentiation direction. So if we look at these 2 directions, the unspliced rates accumulate very fast, spliced rates accumulate very slow. But if you look at these 3 directions, the unspliced rates accumulate very slow, spliced rates accumulate very fast. This indicated this gene are under different regulation mechanism. Maybe the regulation happens during splicing, maybe it happens during degradation, we don't know. But at least they are different. But if you look at the gene expression data, the only information you can get is this gene highly expressed at the end of the differentiation. You will totally miss these 2 information. So this, of course, we predict all the velocity for every gene in this data set, and then we can predict the pseudo time. We can see here is the beginning of the differentiation. Here are 4n. So then I want to go to the next step, because in our algorithm, the unique thing is we will infer every cell's transcription rate, splicing rate, degradation rate. So that is the cell-specific rates. So that will help us to understand the regulation of the mRNA turnover strategy and the regulation of the cell fate. And then, so after we submit the paper, review want us to validate our prediction. So they ask whether your prediction of this rate are correct or not. So we use another case study to validate our prediction. So this case study comes from the embryonic metabolic labeling single cell EU-seq. So basically, in this data set, we will know what is the ground truth of the transcription rate and the degradation rate. We don't have the splicing rate ground truth. So this data was generated from a cell cycle process. So here is the beginning of the cell cycle, and the cell goes from here to here. This is our prediction, and this is the experiment cell cycle position. We can see our prediction is very consistent with the experiment for the cell fate, for the cell cycle. And then we predict the cell cycle gene, the R velocity for all the cell cycle gene. You can see there are many, each row indicated one gene. You can see so here is the unspliced rates, here is spliced rates. You can see so the data is very similar to the Nature paper. So the unspliced rates can predict the spliced rates. And then we predict the alpha and the beta and gamma. So here I show alpha and gamma. You can see this is our prediction alpha, and this is the experiment alpha, and this is our prediction gamma, and this is the experiment gamma. So the experiment value is a little bit noisy than our prediction. I think it's reasonable. And then after we predict the cell cycle, so we want to know whether all the cells, all the genes under the same mRNA turnover strategy or not. So we further cluster genes into different clusters based on the transcription rate and the degradation rate we predicted. We observe something very interesting. For example, in this group, so the transcription rate and degradation rate increasing at the same time, decreasing at the same time. So, but for some of the genes, for example here, the transcription rate is very low, but degradation rate is very high. Transcription rate is very high, but degradation rate is very low. And there are several other groups has different regulation strategy. So let's think about it a little bit. So why a cell want to generate a lot of mRNA fastly and degrade it fastly? So what is the rationale for the cell to do it? So if you want to generate a cell fastly, normally I think it should degrade slowly, right? But there are a group of a group of gene was generated fastly and degraded fastly, and then we look into the function using the geo pathway enrichment. I think it's over here. It's too small. So overall, actually the function is a cell-cell communication. So then it makes sense, because if a cell want to communicate with another cell rapidly, so the gene should be fast response gene. So it should be fast generated and suddenly degraded. So another case study for us is in silico permutation. After we predict all the regulation of a gene and cell fate, so if we ask if we knock down a gene incyclically, whether we can predict the effect on the whole transcriptome. So we tested this function into another data set, is a pancreas development data set. We already know from ground truth, from our pre-knowledge, here is a progenitor cell. It will differentiate into different type of cell, alpha cell, beta cell, I think gamma cell is over here. So here is our prediction. So in this case study, we changed the strategy a little bit to predict the whole field, but not every cell's velocity. In this field, so this field was jointly used by our software and another software published in Cell. So we can in this way we can predict the velocity field. In previous case study, we can predict every cell's velocity, but if here is no cell, we don't know the direction of the differentiation. But for the Rn velocity field, we will know everywhere in the space. So we can see here is the source of the differentiation, here is the terminal position of the differentiation. So all cells come from here and become to this one, this one, this one. Because we already know the cell fate and we searched the literature, we found actually this development was regulated by several very important transcription factors. For example, here's NGN3, PAX4, ARX. So if you want to develop to beta cell, you need to express PAX4. If you want to develop alpha cell, you need to express ARX. So we want to ask, if we knock down ARX, what will happen? And if we knock down PAX, what will happen incidentally? So then we found, because we have the ODE, we have all the parameters, so we can predict the knockdown for every paired gene. So we found ARX and PAX4 are under mutual inhibition. So it is very interesting. If we knock down ARX, PAX4 expression increase. If we knock down PAX4, ARX expression increase. So this conclusion indicated you will never find a cell halfly looks like alpha cell and halfly looks like beta cell, because these key transcription factors are under mutual inhibition. So either alpha cell or either beta cell, you will never find a cell similar to alpha and also similar to beta. After we publish our paper, so we collaborate with several labs. So our study, for example, this is an example. So we collaborate Dr. Sheng at Baylor. So we study the smooth muscle cells differentiation, the regulation of the epigenetic. We predict the cell fate for them, and we publish circulation. And we also collaborate with Dr. Liang Xie, also in Baylor, to study another metabolic regulation for the innate response, immune response. And our sample come from both the mouse model and the patient sample. So here is a summarize. Our tools can be used for the cell fate prediction, and also can be used to predict the transcription rate, degradation rate, and the splicing rate. We can also use our tools to in silico predict the perturbation of every gene. So I want to thank my lab member, Shengyu Li is the first author, and I also want to thank all my collaborators and my funding support. Thank you.
Any questions? No. We still have 5 minutes. Go ahead.
Yeah, Matt, that's just a great talk. So have you would you comment on the possibility of applying methods, for example, to drug-resistant cells? So I also have a small guideline. So we treat multiple myeloma with drug repetitively across maybe 6 months or 12 months, right? And then that cells becomes drug-resistant.
Yeah, it's very important question. Actually, I have several ongoing collaboration try to do that. But we can also collaborate.
Yeah, thanks for the amazing talk. Will you comment on if you think the trained neural network model is good enough, or you think there's still a big room for improvement?
Yeah, I think there is a huge room for improvement because self-fit regulation is very complicated. So in this study, we only use whole genome information, so RNA information. So we didn't integrate the epigenetic regulation like ATAC-seq, and also we didn't integrate the, for example, the microRNA regulation after the transcript, post-transcript regulation. And also this, actually we have several ongoing studies try to upgrade this model to the foundation model using transformer. So we try to collect all the cell-fit related single cell data to train a foundation model that can be applied to any data set, just fine tuning it a little bit.
So the training, during the training, the loss function aggregates over all genes, I suppose. It's not one, it's the same network for all genes,
yeah.
Right? Okay, so if you were to apply a trained model on a cell which is not like any that it has seen so far, do you have a sense of what it will do?
In this study, in the cell dancer version 1, so we will train a net for every data set. So if you have another data set, we will train a network. We will network. Yeah.
I was wondering, like, this is like only the mRNA level. So what about like the translation efficiency? I mean, if you have low the mRNA model, high aggregation rate, the like translation time is quick. So how about like the integrate like proteomics data in the cell level, rather than providing data? Like, will that help you like to improve your model?
Yeah. This is a very good question. Actually, this is another ongoing study in my lab. So we try to integrate the ribosome sequencing data and also using long reads to study isoform level regulation. Sure.
Are you using the time series data to train the neural network?
No, not yet.
So any kind of data?
Yeah, any kind of single cell data.